Assalamu alaikum and a happy new year from the entire team at Elevare. This is Mueid Bhaiya bringing you a little new year gift. So sit back, get comfortable, maybe grab a blanket, and let's make Biology Unit 5 a lot clearer today. In this video, we'll break down the January 2026 Unit 5 article, highlight what actually matters for the exam, annotate the key sections, and go through likely exam questions based on it. By the end, you won't just understand the article, you'll know how to use it in the exam. So, let's get started. Let's start with what the article is. It's basically a text given by your board, Pearson Edexcel, usually a few months before your exam. The good news is, questions from the article cover 20 out of the 90 marks of your Unit 5 biology paper. So, I hope you understand how important this is. How to use the article to prepare for the exam? Number 1, watching this video, right? Going over the text thoroughly, identifying key topics, and reviewing predicted questions. I'll be helping out a bit with this video. It is going to be a long one, so sit back and relax. Watch it over again and make notes. Do the predicted questions. I'll not be giving the answers to the questions. I'll leave it to you to find out so that finding the answers acts as revision. So now let's get to annotating the article part by part, pulling out key ideas and moving into predicted questions across subtopics from the article itself. So here is your article for the January 2026 exam. The exam is 13 days later. So the time you're watching this video, it's going to be the first day of 2026. I hope 2025 was great for you. And we all pray so that 2026 is even better. So let's get started. In the article, the main idea of the entire text, of the entire article that they gave to you is about revolutionizing food using microorganisms or microbial techniques. That's the gist of the entire thing. As they've mentioned here, you'll get questions for question number 9 in the article here. Now, when you get the Unit 5 paper in your hand, there are some students who like to go serially from question 1 to question 9. And there are others who already know that question 9 will be based off of the article and that they can just zoom out through all of the questions. So they go directly to question number 9, finish off those 20 marks, and hopefully you'll get at least 14 or 15 out of the 20, inshallah, if you prepare properly. So in the article, we'll go paragraph by paragraph and then we'll look at a synoptic view as well. They start off with an abstract of what this thing or this entire text will do. So the main gist of the abstract is different applications of microorganisms in food, the use of microbes to produce whole foods, and a lot of ways of how microorganisms can be included in daily food habits, right? So what they do here is they state the problem, how they are targeting it, and methods being used. So like this, paragraph by paragraph, we'll go on with the entire thing. It won't take more than 10 minutes, I hope. So let's move on to the introduction. Now the entire introduction is divided into 3 paragraphs. Paragraphs 1, paragraphs 2, 3, and 4. Here you can pull out a lot of information from here. So they start off by mentioning the crisis in the current food system. Now you could ask why that is happening, and they answer this themselves. This sentence, degradation of land, climate change, and climate disasters, which are set to rise in the future. This is the answer to the question of why the current food system is being pushed to a crisis due to degradation of land, climate change, and climate disasters. Now, why is plant-based system not enough, you might ask? The current diets that we follow are mostly animal and plant-based, right? So here, they answer that question as well because it relies on climate or season and intensive land, water, and chemical use. So this portion says, why is plant or animal based diet not enough? Okay, now they mention another statement, how microorganisms have the potential to overcome this crisis. As I mentioned before at the start, that this entire text is for revolutionizing food with microbial techniques, right? So how are they presenting this idea that microorganisms have the potential? They again answer this themselves. Genetic and system design can advance sustainability. They're highly resilient due to their decentralized nature. Also, they have a high nutritional profile. So here, sustainability, highly resilient, high nutritional profile. So this entire thing from here all the way up to the end of this paragraph, they answered this question. So why are we annotating like this, you might ask? To extract information. If they ask a question in question number 9, let's say a 2-mark question on why do certain scientists or biologists feel that microorganisms have the potential to revolutionize the food industry, you'll have the idea that the answer is in paragraph number 2 in these few lines, right? Because you've read the article, you've analyzed the article enough. And that's the purpose of this video. I hope you can annotate on the article yourself as well. It would be great if you have it printed out on your side as you're watching this video. If you don't, kindly do print it out and then watch this video again. And I hope annotating alongside with me will help you out a lot more. Okay, now let's move on to the next paragraph. This one introduces the concept of using fermentation. Here, fermentation can be used both directly as well as in production to increase the nutritional value of food. So this much is about fermentation. Here is where they introduce the idea of fermentation. They will expand upon this a lot more, as you'll see as we go on to the next paragraph. Next paragraphs, my bad. And they also mention overcoming current limitations using microbial based foods and fermentation. Whenever they mention a statement, they will almost always mention the answer within the same paragraph themselves. So if they mention this overcoming current limitations, they must mention how we are going to do that. So that includes high resolution, high throughput characterization of microorganisms, genetic and metabolic engineering, and selecting strains to improve flavor, nutrition, etc. All of which are the limitations of the current system, which will be overcome by microorganisms. So you'll see this idea repeating itself where they mention a point which is a problem in the current food industry or in the current fooding habits, and then they try to back it up with how microorganisms are going to revolutionize that. You'll see this repeating on and on. Okay, on to the next page. In this paragraph, there's a single important line, and I don't think the other bits are as important. The field of single cell proteins and engineered microbes in food has high potential. They will explore this in this review, so keep this in mind. All right, and this is the end of the introduction. Now we will get into the topics of this article. Right, they will organize the entire thing into uses of microbes over here, and then to the nutritional profile of microbes, and then they'll mention the obstacles and conclusion to finish it off. Have some patience, we'll go through it, the entire thing. Now, I am annotating this entire article, I am introducing questions, I am giving you pointers. You should know that this video is not exhaustive. There is a highly likely chance that there will be questions outside from what I mentioned in this video. I'm just trying to give you an overall idea of how the questions are going to be made so that you can expand upon this yourself as well. Okay, so the rise of fermentation in history. Here, the first paragraph introduces what type of fermented products can be produced, you could say. That's written here. Different fermented products and what those are mentioned in the last 3 lines. There are cheese, yogurt, alcoholic products, fermented bean products, other vegetables, kimchi, etc. All of these undergo fermentation in their preparation process. So, what food is prepared from fermentation? All right. I'm not mentioning the other portions because you'll get an idea of the paragraph if you read it. I'm just highlighting the important bits. In the next one, they mention some alternatives to traditional fermentation. And what are those? They are the rise of plant-based diets, increasing access to international foods, which include traditionally fermented products, and all of this draws examples. For example, the tofu, which is a fermented soybean product from Indonesia and China, which is now used as a meat alternative protein globally, right? So this is a food produced by fermentation, which was produced by fermentation traditionally, but now there may be other forms, such as it can be produced in factories by artificial fermenting agents or whatever they use. So that's the thing to keep in mind. Effects of fermentation. That's written over here. Effects of fermentation. All right. What do? They play an active role in food preservation since ancient times. They inhibit the growth of contaminating or spoiling pathogens and increase shelf life. Some other bacteria or other microorganisms that contribute to fermentation could contribute to nutrition, texture, taste, and smell, right? And depending on their composition, fermented food can bring health benefits. So in this page as well, after all of the information, we go back to the same idea that fermented products are beneficial to health, and this is how the problems will be solved. On to the next page. From here, they start introducing a few pointers on how microorganisms or fermented stuff can be helpful or what sort of benefit we can derive from them. So there's nothing to analyze here. You just need to know what information is in which paragraph. So if they mention the word probiotic, all right, or increasing biological value or of high biological value, it should be from paragraph number 8. All right. If they mention the availability of nutrients, it should be from paragraph number 9. Here they mention another thing, which is a trypsin inhibitor. Now from IGCSE, you know that trypsin is an enzyme. Now if you inhibit enzymes, that's a form of treatment, or it can be used as a form of improving some food digestion or increasing the glycemic index of some foods, right? So that the digestion is better, or the digestion is slower so that the food can be absorbed quickly, etc. They mention that trypsin inhibitors found abundantly in various cereals, grains, and legumes, which are harmful because if you inhibit trypsin, it's going to reduce the digestion of proteins, and if you don't digest proteins, that will hamper the nutritional value that you get out of the food, right? So this is shown of reduced activity in fermented foods. So they're indicating that fermented foods have less trypsin inhibitors, and that is a good thing in the end, right? Now, enzyme inhibitors could be good as well. For example, in some diseases, the medicine used is an enzyme inhibitor. Let's not get into details of that because that's out of the scope of this article. Now, if questions come from the glycemic index, it will be from paragraph number 10. It has low GI foods are beneficial to health. They reduce heart disease risks, etc. And if you see any questions from toxins or toxic substances, it should be from paragraph number 11. Now you have a word consortia here, and you shouldn't leave out any word that is confusing for you from this article untouched. You should know the meanings or the basic idea of all words. So consortia is a group of organisms teamed up for the same goal. Okay, so there can be a lot of scientists which have their separate labs, but they all can be grouped up for the same goal, which is revolutionizing food with microorganisms in this case. Now they mention another thing, which is enzymatically reduced. What do you understand by the word reduced? It basically means the gain of an electron or the loss of an oxygen, right? Now this reduction occurs due to enzyme action, and that's what enzymatically reduced means. And then in paragraph number 12, we see that biochemical pathways produce health promoting compounds, and some of these compounds are antioxidants, some are polyunsaturated fatty acids, some are linoleic acids, vitamins, minerals, etc. Now you could be asked about antioxidants and how they reduce cardiovascular diseases. Remember topic 1 from AS Biology? This is from there. You should understand that Unit 5 is a combination of your IGCSE, AS and A2 syllabus and the entire thing that you've learned all throughout these 2 or 3 or 4 years from school till A levels. So you can expect questions from anywhere in these syllabuses. All right. Now let's move on to the next paragraph. Here we see that they mention a few keywords as well. They mention histamine. You should research what this does. I'll not answer all of the questions, right? So that you have something to ponder upon as well. Sorry about that. Yeah. What does histamine do? That's your homework. All right. Then they mention optimizing strain selection and engineered strains. After you hear these two terms or these two selected word or phrases, you should recall strain production and recombinant DNA. You've learned this since IGCSE. You touched upon this in AS as well. So they could ask questions from here, and it's highly likely that they will mention engineering DNA or producing recombinant DNA in the questions. All right. Now, how this calls for the question as well, where we are asked how differential gene expression occurs. Remember how a single gene can produce different protein or polypeptide chains by RNA splicing, DNA methylation, histone modification, etc. So these are the possible questions that you could extract from paragraph number 13. They also mention trial studies. If you remember trials, you studied double blind trials for medicines. Go over it and review that topic, because that's a very important thing when you're trying to introduce something in the scientific space. You need to go through trials first in the laboratory, and then on test subjects, and then slowly onto humans, and then onto populations, etc. Microorganisms are generally characterized by high protein content. So now we're getting into the nutritional profile of microbes here. So we are first met with high protein content. Now they may ask, what is protein used for? You'll mention protein is broken down into amino acids. Amino acids are used in making DNA, further proteins which are used as enzymes, etc., hormones, other stuff. Now on to the next page. I'm drawing very short summaries out of each paragraph. Your job is to read it thoroughly. I hope you've read it already, because your exam is in 12 or 13 or 12 days. So going over this video, your main aim should be extracting what possible questions could come from each paragraph. So in paragraph number 16, they start off with resistance. Resistant carbohydrates is what they mentioned, but after hearing resistant, the first thing that should come to your mind is resistant bacteria, virus, etc., and how this is brought about by evolution. This is one of the most important topics you could review from this article. One is gene modification, and this one is resistance in bacteria being evolved. That's a common, very common 4, 5, or 6 mark question. So yeah, that's about it. And then they mention health supplements which have immune boosting effects. So if you have the word immune here, you might as well be asked about the immune responses. I have an entire lecture on immune responses summarized into one single class on my YouTube channel. If you want, you could go there and watch it. And then we mentioned polyunsaturated fatty acids. If you remember, saturated and unsaturated fatty acids, polysaccharides, etc. products, you should know the differences between them for the exam. Just go over them, skim over the differences so that you can answer at least 1 or 2 questions based off those. Then they mention the terms endogenous, high endogenous contents. Endogenous means present inside something. So in microorganisms, so this content is present inside the microorganisms. Now, what do they mean by high endogenous contents? They mean the presence of vitamins, minerals, antioxidants, and other functional ingredients. Now on to paragraph number 18. You might be confused about the word elucidated. Elucidated just means explained in simple terms. And here they mention that an elevated RNA content in microorganisms can lead to health issues. Now, elevated means increased RNA content. That can be harmful because RNA is broken down into purine. Purine, and then this purine is broken down into uric acid, which is an excretory product of the body. So if we have very high RNA content, we'll have very high uric acid, which may not be excreted, and then our body will face adverse effects. For example, kidney stones, as they mention. All right. And then we obviously have the phenotypes and genotypes being involved if we are mentioning DNA engineering strains of DNA in the lab, and so on. Now let's move on to the obstacles and future perspectives. They've divided it into technical obstacles, food safety. Under technical obstacles, we have the namely RNA content, which we mentioned before. And then we have cardiovascular diseases, because uric acid in the body is a factor of cardiovascular diseases, renal diseases, which is basically diseases of the kidney. And then that's up to you to study, because you studied cardiovascular diseases in AS, renal diseases or kidney function, glomerular filtration in A2. So in unit 5 itself, right? So go over them and in your own pace, and then make sure that you cover all of this yourself. That's it. All right. Also, they mentioned purine. You could go over the differences between pyrimidine and purine rings in nucleic acids. Just a bit of information which you could skim over, right? All right, now onto the... Here they mention another term, which is envisage, which just means to visualize. I'm trying to cover the confusing words that you might be intimidated by, which is very easy to find. I'm trying to do the easy work here so that the other parts of the revision and the going over the important bits can be done by you in your free time. Since there's very little time till the exams, we have to focus on the high yield information only. Okay, now they mention breeding. You could go over selective breeding programs, right? Using recombinant DNA, vectors. All of these are star mark topics. There's a highly likely chance that you'll get questions from here. Now, there's algal clades. Clades are groups of algae, or algae as you say it, classified based on evolutionary relationships. So yeah, just another word we get out of our way. Now onto the last page of information, right? Food safety. Under food safety, you have obtaining approval of food safety regulatory bodies. So the FDA, EFSA are the main regulatory bodies in the USA, right? We have regulatory bodies in Bangladesh, in every single country, that analyze food products based on different metrics to see whether it's harmful, whether there's actually any nutritional benefit or not as claimed, right? So methods of approving or methods of obtaining approval are include the use of approved organisms and processes, limiting the application to animal feeding, purification of products, and removing foreign DNA and living cells. This is the method of obtaining approval. All right. Before the conclusion, the last paragraph tells us about a summary of the safety of foods which needs to be considered for each different species. So if you make a food product for cats or for dogs or for rabbits or for cattle, and then you make the same food product, a different variation or a different product for humans to consume, right? You have to consider the safety for each and every species of organisms that you're feeding it to. And that's the work of the regulatory body to investigate, right? So, one potential problem that might be faced is the use of endo and exotoxins, which could cause allergic and adverse reactions when ingested. So, yeah. Um, and then we see fermentation technologies being mentioned. I guess I mentioned this in before, but I'll mention this again. In your IGCSE syllabus, you saw fermenters, different technical equipments used in fermentation methods. So go over that as well. Now, concluding the entire article, they mention how an academic publication is how an academic publication moves from being in someone's diary to being published in a journal, right? So what happens here, and it is good to know that a journal goes from journal is peer reviewed and then it is revised and then again submitted for editorial feedback from scientists, from the board, from the scientific bodies of a country or in the entire world in that matter. And then we have acceptance and defending the article. This is somewhat like how PhD doctoral candidates defend their thesis. So, quite similar to that. And then they present this in conferences. And then it's published. So this is basically a flowchart of how that happens. Just for basic information, right? Okay. In the second last paragraph, they mention that synthetic biology and microbial strain engineering broaden the horizons of microbial foods. This is what they expanded upon previously as well, right? That engineering microbial strains and using different artificial processes in biological systems to increase the availability of nutrients, the nutritional value of foods for creating the desired nutritional profiles. So that's basically what the entire article was about. And then we have tackling nutritional discrimination. Nutritional discrimination is something which is prominent in less developed countries where a portion of the population can meet their nutritional requirements while others cannot. And reduced costs of these microbial foods due to increasing production volumes, decreasing costs, optimizing efficiency of the technology, as mentioned here, can aid the development of microbial processes in less developed areas, which will eventually decrease nutritional discrimination. And as they conclude, they mention 2 points. One is sustainability, which you are already aware of, and then ethics, which is a big topic when anything related to organisms being used in scientific processes is mentioned. So the ethical use of microorganisms. This is another HW of yours. Go over this topic at your own time and then make notes or predict questions based off of that. So yeah, that's it for the entire article being annotated. Yeah. Now let's move on to the predicted questions. So I've divided the entire list of probable questions into 9 subtopics. And let me mention right now, I've used AI in making these questions, curating them specifically to the Unit 5 syllabus. Now, if you just put the article and ask the AI to make questions, you won't get what I've got here, because you'll need to give specific prompts, etc. So you're in safe hands basically. This is not random AI generated content. This is targeted for Unit 5 and for your exams specifically. So the 9 subtopics are here. We'll look at each of them and the list of questions separately. Let's go ahead. The first one is article literacy and scientific comprehension. So that's basically whether you've read the article and understood it in its essence, right? So here, let's fix this. And yeah, you could be asked about definitions from the article. As I highlighted some points where they mentioned the point and then mentioned the answer themselves, risks, benefits stated by the authors, those could be asked. Straight extraction plus explanation, yeah. You could be asked about cell factories, which is mentioned in these paragraphs, about how sustainability is maintained or what reasons for sustainability are there in paragraphs 1 and 2, anti-nutritional factors in microorganisms, for example, the high RNA content, right, in paragraph 9, health benefits of fermentation, obviously, we marked that as well, limitations of health claims, and then we have biogenic amines and how to minimize them. This is something which I'll give as a homework to you guys. Okay. And nucleic acid health risks. We mentioned this high RNA to purine to high uric acid, right? So this category basically is involved about the article's literacy or the understanding of your article. I'll not waste time. Let's move on to the next category. Okay. Microbial metabolism, respiration, and fermentation. Anaerobic respiration, ATP cycle, fermentation, all of this I've mentioned before. Kindly go over these from your IGCSE AS and A2 syllabi, so that you have a broad understanding of everything. You could be asked about yeast, about anaerobic respiration from topic 7 in A2, if I'm not wrong, and why fermentation leads less ATP, and then how is fermentation used as a preservation technique. So that's about it. Okay. On to the next category. We have gene technology, synthetic biology, and gene expression. This category is star marked for this session because the entire article mentions this again and again. It's very highly likely that you'll get a question based on genetically engineering DNA strains. Alright. Do come back to this video and comment if you've got that question, as that will help me understand whether my analysis was proper or not. So you could get questions on genetic engineering, PCR, gel electrophoresis, recombinant DNA, transcription factors, engineered strains, synthetic biology, and all of that, right? All of this was mentioned previously when we were annotating the article, and here's the list again. Here's a list of questions that you could get. All right. This covers the topics of gene expression and gene technology. It could be from 4 to 6 marks, 2 marks, 3 marks. They could mention how vectors are used. They could mention how DNA analysis is done, how recombinant DNA strains are produced. There's a broad spectrum of questions that could be asked from here. So it's best that you prepare thoroughly for it. Now the next category is about enzymes and biochemistry of food. As I mentioned, they mentioned the trypsin inhibitors in a paragraph, if I'm not wrong. So the use of trypsin was studied in IGCSE Biology. So it's up to you whether you'd like to revise that bit from IGCSE books or not. I'd suggest to just skim over it. Enzyme specificity, that is active site and substrate and details of this, how pH affects enzymes or the primary structure of proteins, carbohydrate structure, poly, di, and monosaccharides, protein quality, those things, enzyme breakdown. And this is an example of toxins. You don't need this one. That's just how enzymes work. pH reducing this activity. This is stuff that you need to Google yourself. I just have them here so that you have material to research as well. Cellulose and starch comparison, proteins, complete protein or incomplete protein. Complete proteins are those that have all the essential amino acids. Incomplete proteins don't have all of them. That's basically it. So yeah, ignore the syllabuses mentioned here. These cover a lot of other stuff as well. So yeah, let's move on to the next one. In topic 5, category 5, we have human nutrition, digestion, and homeostasis. Glycemic index, research this at your own time. How blood glucose is regulated with insulin and glucagon. I'll mention those here. Kidney function, A2. Nitrogenous waste, the nitrogen cycle, right? Digestibility of food, insulin glucagon interaction, as I mentioned here. GI reduction, diabetes risk, kidney excretion of uric acid or urea, RNA intake. Gout is basically when there's a deficiency of iodine. Digestibility of microbial proteins. All of this is stuff that you need to sit down and research, Google, use AI, use scientific articles on different websites and get a general idea of all of these things. So if they ask you questions from here, so that you can answer it. Okay. On to the next category here, quantitative skills and data handling. This is basic stuff. If you're asked to find the difference between two values, to find the percentage change, to convert energy, or to draw a pyramid of biomass or a pyramid of energy, which you've learned in topic 5 or 6 of A2, if I'm not wrong. And then if they give a graph from the article's topics, right, you will need to analyze or describe the graph or comment on the information. These are basic questions that you've done all throughout your IGCSE AS and A2 syllabi. Energy density of yeast, protein yield, percentage growth increase, protein content comparisons. Your typical graph or numerical biology questions, right? Category 7 is about designing experiments. If you've done Unit 6 properly, you should know how an experiment is designed, how important controls, reliability, qualitative testing, these are when designing an experiment. And as I mentioned, screening and trials, when you have something that you give to a crowd or a population, you're obviously required to run trials to make it as safe for consumption as possible. Now, these are a few questions to keep in mind. Okay. On to the second last bit, ethics, regulation, and food safety. Right. Genetic modification regulation. There's laws in some countries which prevents the use of genetically modified food products. So keeping that in mind. Safety approvals from the food safety boards, keeping that in mind. Risks and benefits, whether the benefits outweigh the risks or not. If we see that the risks outweigh the benefits, then that's a no-brainer, right? So yeah, there's that. And here's the questions that could be included. Genetically modified organisms safety, regulatory approval, food safety of engineered microbes, toxins, allergens, contamination, all of this stuff, right? Now let's move to the last one, synoptic evaluation and essay questions. Synoptic evaluation is looking at the entire article as a whole. What does it state? It states that here's the problem of food or nutrition in the entire world, and here's the solution that we're proposing, which is basically using microorganisms, fermentation techniques, other techniques which use microorganisms to improve or to fix this nutritional problem, right? And synoptic questions are the ones which are longer in marks. It's unlikely that you'll get a 10 or 12 mark question, but you could get a 4-mark question from here about global food security, about nutritional discrimination, about the long-term impact of using microbes, right? About accumulation of microorganisms from one generation to the next, about sustainability, ethics, etc. So here we have a list of questions that you could research. Okay. Now I had a few more topics listed which you could be tested on. So let's see them over here before we end the video. Number 1 is the diagram of a bacteria. Of a bacterium, right? Plus labeling and understanding each one's function. That's basic information. If they ask you to draw one, it's free marks, to be honest. The bacterial growth curve. Why am I mentioning bacteria specifically? Because they mentioned microorganisms over here. You need to know or keep all of the basics about bacteria, viruses, fungi, all of this clear. Then we have genetic modification. We have differential gene expression. And some details about digestive enzymes from IGCSE. You had an entire chapter on digestive enzymes in IGCSE. Skim over it and you'll get the basic idea. And lastly, we have DNA sequencing, engineering, plus analysis techniques, PCR, gel electrophoresis, etc. GE is for gel electrophoresis. So these topics are the most high yield that I could find, alright, from the article. And that's basically off of my understanding and my experience with past papers, with teaching students, which is of about 3 years, not that extensive, but I hope you'll find this helpful in your exam. So with this, the entire video comes to an end. Thank you so much for watching till the end. If you've watched till here, I hope it was helpful to you. I hope you could derive information from the video that I wanted to give. There might have been mistakes in the information. Kindly mention them in the comments and I'll try to work on them from next time. Before I end the video, I'd like to say there's not a lot of days before your exam, about 12 or 13 days. Make sure that your mental health, your emotional health, and your physical health are all in check so that you can give your best in the exam. Try to get enough sleep, proper nutrition, right? It aligns with the purpose of the article as well. Get proper nutrition, eat properly, and try to exercise or put your body into some sort of physical activity so that you're not just nerding out on the table, right? So yeah, if you understand the article the way we broke it down in this video, you'll be answering the questions from it with full control and confidence, inshallah. So this is Sheikh Mueid from Elevare signing off from this video. Best of luck for your Unit 5 exam and a Happy New Year again. Assalamu alaikum.